everybody, welcome back to next episode of Scientists on Board. In today's episode, we'll be featuring 4 special guests that will be telling us about DNA cloning process in an animal. If you want to know the process and what animal is it, let's keep on watching. Alright, so before we proceed to the process of DNA cloning, I will first tell you about the uses of DNA cloning. Firstly, in the biopharmaceutical industry, where the production of medicines are made, insulin and human growth hormone are made by the help of DNA cloning. The second use is in gene therapy, where it provides a normal copy of gene to be inserted into a cell of a patient that has a genetic disorder's body. And last but not least is gene analysis. It is use of DNA cloning to build artificial gene so that the scientists can know how does a normal gene function in an organism.
Finally, after hours of fishing, I finally got my fish!
In this video, we're going to learn all the steps that includes in cloning a fish. So, let's move on to the first step.
The first step in DNA cloning that we call as the cutting and pasting of DNA. So our goal in cloning is to actually insert a target gene, which in this case, the fish gene, into a plasmid using a carefully chosen restriction enzyme. This is a bacterial plasmid, while this is the fish DNA. This is the gene of interest or the target gene that we're going to insert into the bacterial plasmid. Using =R1, we're going to cut the fish DNA to obtain the target gene fragment, which has a cut site near each end. Using the same restriction enzyme, the bacterial plasmid is cut. The next step is we are going to combine the fragments with DNA ligase, which links them to make a recombinant plasmid containing the gene of the fish. This is now the recombinant plasmid with the fish gene.
Okay, after cutting and pasting, next I will show you the second step, which is bacterial transformation and selecting. Okay, this one that we want is plasmid and it has green fish DNA insert. But we also get things like this. We will get plasmid that had other part of the fish genome inserted into it. And we also get plasmid that just close back up on themselves. After have new plasmid, we must split them into some bacteria. Here we are going to take some E. coli. As you can see, it already has some DNA inside. The important things about this that they will actually die if we add the tetracycline antibiotic. So what the Boyer and Cohen did to the guide plasmid inside of this E. coli. So they cool all the cell down and then quickly heated them up, creating little holes in the cell membrane. Then the plasmid could slip through and get into the cell. These are the kind of bacteria that we have now. This one is the bacteria that got the plasmid we want. Then, plate all the E. coli that we transform into our new plasmid onto the plate which has tetracycline in it. Then, let the plate into the incubator overnight and allow the bacteria to grow.
Moving on to the production of protein in fish. This involves the process of transcription and translation. For the transcription process, generally in the fish DNA it contains fish gene. When the gene is switched on, an enzyme called polymerase attaches to the start of the gene. It moves along the DNA making a strand of mRNA out of the 3 bases in the nucleus. The DNA code determines the order in which the bases are added into the mRNA. To get the translation started, mRNA attaches to the ribosomes and start coding a strand. The DNA then brought the amino acids to the ribosomes. 3 bases are read at a time by the mRNA. Proteins keep growing until the stop codon is reached. Once it is read, the protein formed and ready to finish folding to become functional. And that's the end of the protein production.
So, you have already listened to all the steps in cloning. We hope that this video helps you understanding more in cloning of fish. I think that's all from us. Thank you for listening and thank you for learning your English.
Yum, yum, yum!